Non-membrane bound ribosome, nucleolus, centrioles, single membrane, Golgi, endoplasmic reticulum, vacuoles, vesicles, lysosome, pili, fimbriae, peroxisome. Double membrane, mitochondria, plastid, chloroplast, nuclear membrane. In eukaryotics, it is extension of cell wall, but in prokaryotics, we use cell membrane. Cell membrane. Cell structure is easily observed. Cell structure is easily observed under light microscope, but its detailed structure was studied only after invention of electron microscope. Chemical analysis of cell was done by okay RBC. Why RBC? No cell organelle. After chemical analysis, RBC has no cell organelles. After chemical analysis, this process of study of physiochemical properties of the cell is called reductional biology. After chemical analysis, RBC, chemical analysis, composition lipid 40%, protein 52%. Lipid 42%, lipid 40%, protein 52%. On the further chemical analysis, lipid, phospholipid, main, cholesterol, and okay, protein on the basis of age of excretion, peripheral protein, and integral protein. Integral protein further buried, transmembrane, partial buried. Glycoprotein, glycolipid, role of cell membrane, cell division, secretion, forming intercellular connection, transport, main role. Okay. Phospholipid 40%, phospholipid and cholesterol. Phospholipid mean and cholesterol animal cell protein on the base of S of excretion, peripheral protein and integral protein. Integral protein fully buried, transmembrane partial buried. Glycoprotein, glycolipid, role of cell membrane, cell division, secretion, forming intercellular connections, transport main role. Bulk, endocytosis, specific, exocytosis, solid, phagocytosis, liquid pinocytosis. Okay. Role of cell membrane, cell division, secretion, forming intercellular connection, transport. Main role. Role of cell membrane, main role hold transport. Then cell division, secretion, forming intercellular. Okay. Specific, endocytosis and exocytosis. Endocytosis solid হলে phagocytosis আৰু liquid হলে pinocytosis. Endocytosis and exocytosis. Endocytosis solid হলে phagocytosis আৰু water হলে liquid হলে pinocytosis. Specific transport. Gases of CO2, O2, water, non-polar molecule like hydrophobic, lipid soluble molecule, polar molecule, hydrophilic, water soluble. Okay, specific transport. Specific transport. Gaseous CO2 and O2, water, nonpolar molecule, hydrophobic, lipid soluble molecule, polar, hydrophilic, water soluble, nonpolar, lipid soluble, and polar, water soluble. Gas, gas pressure gradient. No, okay. Gas through pressure gradient diffusion. Non-polar hydrophobic liquid solution through diffusion. Water through osmosis. Water moves from dilute to concentric. Special case of diffusion, osmosis. Polar or hydrophilic water soluble transport across the plasma membrane with help of carrier protein. Gas. Gas through diffusion. Pressure gradient. Due to pressure gradient. Non-polar or hydrophobic through diffusion. Water osmosis through osmosis. Special type of diffusion, that is osmosis. Polar hydrophilic water soluble transport across plasma membrane with help of carrier protein. Transport across with the help of membrane. Transport across the plasma membrane with the help of carrier protein. Transport across the plasma membrane with the help of carrier protein. Okay, channel passive transport, no ATP. Active transport, ATP utilize. Channel passive transport, no ATP utilize. Pump against the concentric gradient, active transport, and ATP utilize. So, Roll up cell wall in eukaryotic cell wall rigid shape of structure support prevent over expansion of cell, prevent entry of macromolecule, prevent infections, helping interaction between cells. Cells wall rigid shape and structural support over expansion of cell, osmotic bursting, prevent the entry of macromolecule, prevent infection, helps in interaction between cells, helps in interaction. Mm. Bryophyte, pteridophyte, gymnosperm, terrestrial, algae, cell wall. Okay, man and galactans. Okay. Specific transport, some gas like CO2, H2O2, water, non-polar, polar, non-polar are soluble in lipid, polars are insoluble, soluble in water. Okay. Role of cell wall in eukaryote cell wall rigid shape and structural support prevent over expansion of cell osmotic bursting, prevent the entry of macromolecules, prevent infections, help in interaction between cells. Cellulose matrix, Menens galactase. Okay. Middle lamella present between two adjacent cells, act as glue holding the two cells together, made up of calcium pectate. Cytoplasmic connection between two plasmodesmata transfers through the middle lamella. Endoplasmic system, group of organelles working in a coordinated manner. Okay. Got the power. I got the power. Now I'm going to reading NCERT. Okay, eukaryotic cells. The eukaryotes include all the products, protists, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possesses an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structure. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plants and animal cells are different as the former possesses cell wall, plastid, and large central vacuoles, which are absent in animal cells. Plastid and large central vacuoles, which are absent in animal cells. On the other hand, animal cells have centrioles, which are absent in almost all of the plant cells. Centrioles, cell wall, plastid is present in centriole. Let us now look at the individual cells organelle to understand. Okay, cell membrane. The detailed structure of membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells, enabled the scientists to decode the possible structure of plasma membrane found in both prokaryotes and eukaryotes. These studies show that the cell membrane is mainly composed of lipids and protein. Lipids 40%, protein 52%, and other 8%. The major lipids are phospholipids and cholesterol in animals. Bilayer. The lipids are arranged within the membrane within the polar head towards the outer side and hydrophobic tails towards the inner side. The lipids are arranged within the membrane, with the polar head towards the outer side, polar heads towards the outer side, and non-polar tails towards the inner part. This ensures that the nonpolar tail of saturated hydrocarbons is protected from the aqueous environment. In addition to phospholipid, membrane also contain cholesterol. Later, biochemical investigations clearly reveal that the cell membrane also possesses protein and carbohydrates. The ratio of protein and lipids varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein and 40% lipid. Depending on age of excretion, lipid can be divided on two types, integral and peripheral. Peripheral protein lie on the surface of membrane. Peripheral protein lie on the surface of the membrane, while integral protein are partially or totally buried in the membrane surface of the buried in the partially or totally buried in the. This is sugar, this is peripheral protein, integral protein, phosphobilayer, okay, cholesterol, sugar, peripheral protein, integral protein, phosphobilayer. Rough endoplasmic reticulum, lysosome, nucleus, nucleolus, Golgi apparatus, nuclear envelope, plasma membrane, vacuoles, middle lamellae, cell wall, mitochondria, ribosomes, chloroplast, cytoplasm, peroxisome. Okay. Microtubules, plasmodesmata. Okay, plasmodesmata, smooth endoplasmic reticulum, rough endoplasmic reticulum, lysosome, nucleus, nucleolus, Golgi apparatus, cell envelope. This is plasma vacuole, cell wall, mitochondria, ribosomes, chloroplast, cytoplasm. Okay, peroxisome. It is peroxisome. Um, and improved model of structure of cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of lipids enables lateral movement of. Okay, according to this, the quasi-fluid nature of lipid bilayer enable lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. This ability to move within the membrane is measured as its fluidity. The fluid nature of membrane is also important from the point of view of function like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of molecule across it. Membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Channel, channel or pump. Neutral solute may be moved across the membrane by the process of simple diffusion along concentration gradient, that is, from higher concentration to lower. Water may also move across membrane from the higher to lower concentration. Movement of water by diffusion is called osmosis, as the polar molecule cannot pass through the nonpolar lipid bilayer. They require a carrier protein, as the polar molecules do not cannot pass through the nonpolar carrier protein of the membrane, facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentrated carrier. That is active transport. ATP utilize sodium potassium pump. Passive transport channel and active transport sodium potassium pump. Cell wall dead. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of the fungi and plants. Cell wall not only gives shape to the cell and protect the cell from mechanical damage and infections, it also helps in cell-to-cell interaction, provide barrier to undesirable macromolecules. Algae have cell wall made of cellulose, galactose, manganese, minerals like calcium. Okay, algae have cell wall made up of cellulose, galactose, manganese, and minerals like calcium carbonate. While in other plants, it consists of cellulose, hemicellulose, pectin, and protein. Consists of cellulose, hemicellulose, pectin, and protein. The cell wall of a young plant cell, the primary wall is capable of growth, which gradually diminishes as the cell matures, and secondary wall is formed on the inner side of the cell. The middle lamellae is a layer mainly of calcium pectate which hold up or glue the different neighboring cells together. The cell wall middle lamellae may be transversed by plasmodesmata which connect the cytoplasm of neighboring cell. Okay. Middle lamella is layer mainly calcium made up of calcium pectate which holds or glue the different neighboring cell together. Connect the cytoplasm. Okay. Middle lamella glue or hold the two different cell together, two different neighboring cell together, and plasmodesmata connect neighboring cytoplasm of neighboring cells. Okay, holes to different neighboring cells are plasmodesmata connect the cytoplasm of two neighboring cells. Plasmodesmata connected cytoplasm of neighboring cells. Algae cellulose. Okay, cellulose, magnan, cellulose, galactans, manganese, and minerals. Cellulose, minerals like calcium carbonate, manganese, galactans. Okay. Water molecule through osmosis, non-polar molecule, and gases through diffusion are with the help of carrier protein. Passive transport channels, no ATP utilized, are active transport, sodium, potassium, pump, ATP utilized. Now I'm going to solve my modules questions.
as well as glycoprotein. आपको glycoprotein दिखेगा, glycoprotein मिलेगा. Glycoprotein क्या है? शुगर प्लस प्रोटीन. शुगर प्लस प्रोटीन आपको लगा दिया तो आपको क्या मिल जाएगा? glycoprotein. और अगर शुगर प्लस लिपिड लगाया तो आपको क्या मिल जाएगा? glycolipid. ठीक है? So basically this is the ultra structure of your plasma membrane. जितना भी क्वेश्चन आना है इससे क्वेश्चन आना है. ठीक है? राइट? नेक्स्ट आपको ये याद रखना है कि मैंने वहां पे भी दिखाया था कि क्या होता है मॉडल ऑफ प्लाज्मा मेम्ब्रेन से आपका क्वेश्चन है. अभी मॉडल ऑफ प्लाज्मा मेम्ब्रेन डायरेक्ट स्टेटमेंट क्वेश्चन. मॉडल ऑफ प्लाज्मा मेम्ब्रेन आपको क्या समझ आता है? मॉडल ऑफ प्लाज्मा मेम्ब्रेन आपको समझाता है अरेंजमेंट ऑफ लिपिड्स एंड प्रोटीन्स. क्या समझाता है? अरेंजमेंट ऑफ लिपिड्स एंड प्रोटीन्स. सो अरेंजमेंट ऑफ लिपिड्स एंड प्रोटीन्स समझाने के लिए मेरे को बेसिकली बहुत सारे मेम्ब्रेन थे, मेम्ब्रेन मॉडल थे, जिसमें से सबसे इंपॉर्टेंट वाला कौन था? सिंगल एंड निकोल्सन. सिंगल एंड निकोल्सन का मॉडल का नाम क्या था? फ्लूइड मोज़ेक मॉडल. किसका था? फ्लूइड मोज़ेक मॉडल. ठीक है? अभी अकॉर्डिंग टू सिंगल एंड निकोल्सन फ्लूइड मोज़ेक मॉडल है, राइट? नाउ फ्लूइड मोज़ेक मॉडल says the fluid mosaic model says that our plasma membrane is quasi fluid. ये quasi fluid है, डायरेक्ट स्टेटमेंट है. plasma membrane is quasi fluid मतलब ये neither solid nor liquid. बोल लेते हैं. ठीक है? neither solid है nor liquid है. ठीक है? राइट? एंड the fluid mosaic model the plasma membrane shows membrane fluidity. membrane fluidity दिखाता है. तो what is membrane fluidity? membrane fluidity वो लोग ये बताते हैं कि जो प्रोटीन थे ना, the protein को lateral movement move कर सकता है. suppose this was the protein. this was the protein. ये प्रोटीन अगर चाहे तो ये चल के इधर भी आ सकता है, ये चल के वापस इधर भी आ सकता है. lateral they can move lateral. ऐसे ऐसे मूव कर सकते हैं, यहां से यहां lateral movement कर सकते हैं. so because they can move at a free will. ठीक है? तो ये क्या बताता है? ये हमको बताता है कि the membrane shows fluidity. two statements है कि the plasma membrane is quasi fluid, right? it is neither solid nor liquid, and the protein क्या दिखाता है? lateral movement. that is why the membrane shows fluid. इसके अलावा हमारे बुक में नहीं है, लेकिन आपका ये जो लिपिड है लिपिड बाइंडर